Hello, I'm Renzo Huber, and I want to thank the organizers for inviting me. It's really a pleasure, and I have no financial interests to disclose with respect to this presentation. And I'm really excited to kick off this functional MRI part as part of this ultra-high field workshop, because I think fMRI is important for ultra-high field imaging as it is the other way around. At conventional field strength with conventional resolutions, fMRI already has taught us a lot about the big structures in the brain, like how vision is involved back there, where we move, where it's represented, how to move the body, listening, and also uniquely human abilities like language. However, as exciting as it is to see the brain in action like that and it develops tools for that, it's sometimes frustrating how little we learn about the mechanics of the brain by looking at these kind of fuzzy fMRI blobs. It's somewhat comparable as trying to understand how a truck engine works by looking at it with an infrared camera. We only roughly see which kind of big structures consume energy and are heating up, but we learn very little about the kind of densely packed microcircuitry. We don't get a mechanistic view. So at ultra-high field accessible resolutions, we do not only see gray matter, but also activity distributions within gray matter, aka across the cortical layers. And these different layers have different neurons that do different things of neural processing. So going from conventional resolutions to ultra-high field accessible resolutions gives us a lot of new information to work with. So we not only learn if a brain area is involved in a certain task, but also where this activity comes from, how it's following the different processing steps, how it's modulated by other input, and until the result of the processing is sent to further brain areas with respective feedback and so on. So we not only learn which brain areas are connected to each other and which of those connections are stronger than others, but also we learn something important about the causality, the directionality of these information flows. And that's a very valuable information to have. And because this information is so valuable, the field of human layer-dependent fMRI is really exploding at the moment. There are almost 200 papers published to date, and obviously I cannot give all of them justice in a 20-minute presentation, but still I want to give you a feel about the field of human layer-dependent fMRI by showing you some statistics of all of those papers. Here, for example, you can see that it took quite a while from the first proof of principle studies about 25 years ago until it really took off in 2015. And it's not that nothing happened here. I think this was the golden age of understanding the contrast mechanisms and like neurovascular coupling mechanisms, which was largely conducted in preclinical studies. So currently the field of human layer-dependent fMRI is standing on the shoulders of giants, of these animal researchers doing the groundwork. Layer fMRI is really mostly done at 7 Tesla, and I think it's one of the reasons many 7 Tesla scanners are being bought by centers. It's mostly Siemens for that matter. And since you're attending an ultra-high field workshop, you'll know that there are about 102 ultra-high field scanners installed worldwide. And about a third of them is located in Europe. And there's this weird European bias in the sense that 2/3 of these layer-dependent fMRI papers are using the European sites. And these are the same data just visualized differently in order to show you how many labs there are that do layer-dependent fMRI. It's quite many. Until about 2018, the field was really focused on methods and method development papers. After 2018, there are more application studies, mostly looking at vision compared to method studies. So nowadays, the neuroscientists really are taking off, and the average layer-dependent fMRI researcher is no longer attending these kind of technical workshops like this one. There's quite a bad gender balance in our field. 3/4 of the authors are male, and when you look at the references within layer-dependent fMRI papers, only 1 out of 7 references goes to a female authored citation, which is really a bias that we cannot be happy about in our field. Just like conventional fMRI, also for layer-dependent fMRI, the most commonly used sequences are gradient echo bold contrasts. About half of the papers use it. The other half also uses additional other sequences on top of that. And the question of the most suitable layer-dependent fMRI sequence is a question that really got a lot of attention in the last decades, maybe distractingly much so. So when I entered the field a while ago, I perceived the spectrum of available methods as kind of being part of this epic battle to find the ultimate winner, and there can only be one winner that is the best layer fMRI sequence out there. And I understood many papers as saying that there's this one superior method as opposed to another one. with very little generalizability beyond a very specific experimental setup for where these comparisons were conducted. And definitely very little consensus in the field. So I think nowadays the field kind of shifted more to this kind of trade-off model where you compare different sequences like you would compare different vehicles based on a number of quality features. Like an airplane is very fast, right, but maybe less comfortable. And gradient echo bolt might be super sensitive but less specific. And then depending on the requirements of your experimental setup, you might use one or the other. Like when you're on a lake, you use a ship. For your commute, you use a car. And when you're sensitivity limited, you use gradient echo bolt. When you're specificity limited, you use something else. Which is a kind of more healthy way to think about it, though I doubt that this will be the end of the discussion yet, because it still implies that for a given experimental setup, there's one ultimate best method to use, which I think is hard in the sense that sequences, like vehicles, are 100% man-made engineering products. They're not mysterious phenomena of Mother Nature that you can do science on, right? You can never disprove any of it. Like I put comfort very little for an airplane. But have you tried first class that I can't afford? Or I put field of view very low for VASO, but have you tried the last method that we haven't released yet? So I think in the future, the most powerful studies will not use one method, but will use a combination of methods and then benefit from the best quality features combined. Similarly, on my way to get here, right, I didn't walk to the airport. The most efficient way is to use a combination of methods. Just like for conventional fMRI, also for layer-dependent fMRI, we usually need to find a good compromise between spatial resolution, coverage, and temporal resolution. And you can push either one of those parameters by making sacrifices with the other ones. And I would like to talk about challenges in either of those in the next few slides, starting with spatial resolution. And when it comes to spatial resolution, I'm particularly excited about the development of the Fine-Bergertron scanner, which is really a scanner dedicated for layer-dependent fMRI. And we will hear more about it tomorrow from David. And that scanner is currently equipped with a 96-channel coil from Jajan. It has this impulse gradient built from Siemens, co-designed with MGH. And David and his team set up this very convenient remote scanning system and allowed me to use that for more than 10 sessions already in order to explore the resolution limits of layer fMRI VASO with these segmented approaches from Rüdi in Bonn. As a visual guidance here, I'm zooming into the primary visual cortex and highlight these large draining veins, and maybe unsurprisingly, those are the hotspots of gradient echo bolt. However, at the resolution here of 0.39 mm isotropic, we are entering uncharted territory, and we don't really know what to expect. And if all the kind of vein biases at lower resolutions still apply here. In fact, not everything that looks like a vein has a huge vein bias in there. So the plot thickens about this infamous vein bias, because I do think that at these high resolutions, the intravoxel defacing is just different than at lower resolutions. More interestingly, maybe looking at the blood volume VASO, we are starting to Nyquist sample different layer groups at these resolutions. For example, layer 2-3 different from layer 4C with a tiny gap in between even. Which is something that so far I've only seen in monkey data from Josin Gönze, for example. Not even in rats where layer 2-3 are very, very thin. But also there we do see surprising things, unexpected, that these kind of double stripes come in this interdigitated pairs of hotspots, where we are still debating what this might mean. In any case, I think for the field it's really exciting now to get access to these resolutions and see things that could not be seen before, and then as a field we have the fun task of solving the puzzle to figure out what's real and what isn't. When it comes to coverage, I think the field really has come a long way, in the sense that just 5 years ago it was completely normal, for me at least, to look at individual brain areas with like not even 1% brain coverage, like the primary motor cortex as shown here. But with efficient segmentation and acceleration methods, you can nowadays basically choose any matrix size that you want and then just stack slices linearly until you end up with something like whole brain at the cost of TR. And one of the whole brain projects for layer-dependent fMRI that I'm particularly excited about is also presented in poster 33 from Kenju. Years that I struggled with artifact-infested readouts, he now found a nice way to use the same undersampling pattern, in fact, but just change the trajectory in order to keep the shot-to-shot B0 variations completely orthogonal to the low bandwidth directions, giving us quite consistent TSNR, and also the inherently nice T1 contrast of VASO allows very high-quality segmentation in the distorted DPI space. Still, it takes weeks of manual labor and correcting free surfer outputs, but at least then you know that you have the right results in the distorted DPI space. Kenjo uses these movie watching tasks, namely the HCP movies, using a collection of 6 movie clips together about 15 minutes and shows it 51 times. And here you can see the excellent test-retest reliability across days here. But Kenjo does not only look at univariate analyses, but also pattern analyses, specifically representational dissimilarity analyses. And this means that for each movie frame, he extracts the spatial layer fMRI activation pattern and looks at how similar this pattern is compared to all the other movie frames, giving us these kind of representational dissimilarity matrices where you might see some structure. If you squint your eyes a bit, you definitely see kind of areas that are more dissimilar compared to other ones, which might be better visible when you average within the movie clips. For example, here that these kind of nature scenes are very, very similar compared to the beach scenes from the Bahamas, whereas this guy talking compared to these object viewings are more dissimilar. And the convenience of working with these kind of representational dissimilarity matrices is that you can directly quantitatively compare it with behavior. Just asking the person, is this pair of movie clips more similar than that pair of movie clips? Then you rank it and you get the same matrix of the perceived similarity as opposed to the brain representations, where we do see that some movie clips are just consistently more similar compared to other ones. And this correspondence is highly dependent on areas and also layers. For example, this nice correspondence specifically visible in kind of high-level visual object recognition areas, feed-forward driven, as opposed to feedback-driven frontal areas. When it comes to temporal resolution, I would like to refer you to the work from Sebastian Dresbach, poster 41. If you haven't checked it out, do so. He uses sub-second readouts in order to explore the usability of event-related compared to block designs for layer-dependent fMRI using the somatosensory visual protocols here, where he does see clear delays across cortical depths. For example, that the superficial layers in blood volume vaso are almost 2 seconds delayed compared to the deeper ones. So check out his poster in order to see if and how layer fMRI can be used for these faster neuroscience-driven tasks too. When you want to push it even further, you can also combine layer-dependent fMRI vaso with spiral readouts, here presented from Denis Courban, looking at SMS spirals, and Alejandro Moreal looking at 3D stack of spirals. And both of those approaches use these kind of Kaipi blips continuously during the readout while the ADCs are open, which makes it a kind of 2D collapsed wave Kaipi approach, if you want to think about it that way, with clear benefits in SNR. And like, it's so efficient, it doesn't really get much faster than that. And while the resolution is just not quite there yet to look at layers, this is definitely something in the very near future that both projects want to look at. Coming back to this triangular parameter space, I think it always comes down to SNR. And if you would have more SNR, you could push all of those parameters at the same time. So I think in the thermal noise-dominated regime of layer-dependent fMRI, we might particularly benefit from these denoising tools like Nordic. And this is a slide that you might see again in a few minutes from Luca that really highlights the power of these approaches, turning this kind of salt-pepper noise here into nice activation maps with very reasonable time courses too. And it's particularly impressive because this is also 0.4 millimeter resolutions at the 10.5. Aside of all the acquisition challenges, I think layer fMRI analysis is similarly important. So Farouk and I are trying to provide a dedicated layer fMRI analysis suite called Laney, which is a C++ suite, open source, to completely work in voxel space. And the necessity for such a layer fMRI dedicated software suite comes from what I call the beer-wine dilemma. So conventional fMRI, representing the beer here, has been standardized over decades to give you data that can be homogeneously pushed through standardized pipelines like done by international brands of beer corporations. Layer fMRI, however, representing the wine here, is not so standardizable just yet. It's messy, maybe like ultra-high field in general. And we are dealing with data that have study-specific artifacts, and you cannot easily push the data of one study through the analysis pipeline of another study that had different artifacts. So just like wine, you do get the best results by not pushing the data through Henry Ford-like assembly line production, but instead give the user a lot of flexibility and even parameter tweaking, which has the limitations that it's not strictly following the trends of how current reproducibility standards are being understood, in the sense that in layer fMRI you cannot easily standardize without a reduction in quality. And in fact, we try to increase the reproducibility by individualized focus on quality. And one of the most important implementations of LANI is the layerification in small field of view voxel space. Here, for the sake of validation, shown in a cadaver brain visual cortex, where you can see the tree of Gennari and how the different layerification algorithms nicely follow it, specifically the equivolume approach. And the flexibility of LANI can then be seen in the sub-microliter Weinbergertron data, which can be messy in the sense that we have multiple sessions, partly overlapping slabs, and you need to do the segmentation directly in the gradient nonlinearity distorted EPI space. So we definitely do not fulfill the topology requirements of mesh-based layering, having closed surfaces without holes, right? But in LANI, it works in voxel space. We don't care about topology requirements. It gives you layers out of it, just representing your segmentation quality, with or without topology. In cases where you can assume that neighboring columns are doing the same thing, same processing, you can apply layer-specific smoothing without signal leakage across cortical depth in order to view data qualities also in noise-dominated individual runs. LANI also does topographical analyses, here again shown in a small patch of the cortex, where you just pick any seed region, and then LANI spans the local coordinate grid, and then you use that in order to flatten the cortex and look at columns and layers completely orthogonalized, again in 3D in Nifty. You give LANI a Nifty and you get out a Nifty, and everybody knows how to view and manipulate Nifties, which can be a very powerful viewing tool that informs many studies. Here, for example, this study, where Silvina and I are looking at how usable layer-dependent fMRI can be to inform neurology, specifically looking at focal hand dystonia patients, where independent of the patients and hemispheres, we do get quite consistent blood volume results for these individualized finger tapping tasks. And then spanning this local coordinate grid, you can see how in the affected and the unaffected healthy hemispheres, the finger representation nicely follows this kind of supanotopic pattern. Whereas in the affected hemisphere, not for the lack of sensitivity, it's completely messed up. There's no clear structure there. And maybe more interestingly, looking at the layer domain in the primary motor cortex, you can see that for the affected hemispheres, for vaso in blue, we have this clear bias towards layer 2, 3 as opposed to the deeper layers, which is not visible in the unaffected hemispheres for vaso in green. And this is very interesting because one of the theories of focal hand dystonia predicts such a superficial bias due to the lack of surround inhibition. So our data clearly focus or clearly favor this theory about the disease. Another very exciting application of the patch flattening in LANI is looking at orientation selectivity, aka these axes of motion in human MT-V5, where the orientation selective voxels nicely follow these kind of columnar patterns, as you can see here, with very few exceptions where it doesn't really look super columnar. And the different colors refer here to the different orientation, nicely interdigitated, with improved orientation selectivity for vaso. So stay tuned for Alessandra's talk coming up in a few minutes. Layer fMRI, and specifically layer fMRI vaso, has been conducted in many cortical areas already, and one of the hardest cortical areas still is the primary auditory cortex, where the kind of cardiac pulsation artifact with relatively long 3D EPI readouts just basically make the Herschel gyrus disappear. So Lonique Fais here really had to reinvent the wheel on so many acquisition choices, looking at Grappa reference schemes, looking at inflow effects, looking at 2D versus 3D, until we ultimately ended up with a quite nice, concise 3D small field of view acquisition blob following a silent gap that we need in vaso anyway to present stimulus and then amplify the response, which gives quite consistent results across participants here. So we do see that in vaso we definitely benefit from 3D acquisitions, and in gradient echo bold mostly from 2D SMS. And we do see this reduced domain bias in the superficial layers as expected. Currently, layer-dependent fMRI as a field is growing faster than the ultra-high field community. So in order to keep growing, I think we do need to expand across field strength. And while ultra-high field strength, I think, should stay in the leading role of being a technology driver, conventional like 3 Tesla field strength also have quite a few virtues, in the sense that there are orders of magnitude more users that could cite your papers and use your tools, but also the longer T2 star and reduced B0 inhomogeneities make it so much easier to work with and do all kind of segmentation tricks here. Together with Martin Kronbichler, we looked so far at visual protocols, motor protocols, and whole brain movie watching tasks, which provide quite reliable data quality if you're willing to scan a bit longer. And in Maastricht, we have a quite wide spectrum available of field strengths, so we can also nicely compare the efficiency across field strengths in the same participants, where you do see that the results are quite consistent. And maybe again highlighting the convenience of using a sequence that gives activation results in physically meaningful units, namely milliliters, so you can compare it. For further developments in the field of layer-dependent fMRI, check out the latest special issue in Progress in Neurobiology. And with this, I thank all my colleagues and I thank you for your attention.